Hello guys, I'm back again and today we are talking about mitosis and cytokinesis. First, what is mitosis? Mitosis is when one cell divides into one cell divides into two daughter cells which are identical to each other. So what is the purpose of mitosis? Well, the purpose of mitosis would be, of course, healing, growth, replacement of dead and dying cells, like you want a skin cell replaced by a skin cell, right? You don't want any bone cell, etc. So once again, a mother cell produces two identical daughter cells. All right, the cell cycle has four main stages. So what is a cell cycle? A cell cycle is a regular pattern of growth, DNA replication, and cell division. So a typical cell cycle will have the longest phase, which is called the interphase. Interphase has G1, S, and G2. G1 is gap 1. The cell in this phase will grow, carry out normal functions, and replicate the organelles. The S phase is synthesis. Here DNA synthesis takes place so that the chromosomes now are duplicated. All right. And here is G2, which is the additional growth. This is when the cell will make necessary proteins which are needed for cell division, like cell membrane proteins, cell wall proteins, if it's a plant cell, etc. Now comes the M phase, that's the mitosis. Mitosis cell division, notice, is not as long as the interphase. So cell basically will grow, become of a certain size, then only it will divide into two identical daughter cells. That is basically what the cell cycle is all about. All right. One more thing I need to tell you is that there are two checkpoints between G1 and S and G2 and M phase. And until and unless you do not have these checkpoints are not clear, then only the cell will go from G1 into S. Otherwise, it'll stop. So for a cell to go into mitosis, remember, the DNA has to be totally undamaged. It has to be, everything has to be duplicated and ready to go. And then only it will go into the M phase. So mitosis is divided into four phases: prophase, metaphase, anaphase, and telophase. We are looking at the into the first phase, that's the prophase. So in the prophase, the first thing that you notice is the chromosomes are condensed, and you notice that they are duplicated. So here is one sister chromatid, here's another sister chromatid, and this point, what it is joined in, this point is called the centromere. Okay, and these are sister chromatids. So one sister chromatid, another sister chromatid joined. So you will notice that the chromosomes that are duplicated in the interphase, in the S phase of interphase, now they show up as condensed and they're visible. You can count them. Next, nuclear membrane. So number one is this. Number two, nuclear membrane is going to break down. Notice that. The centrioles also, what you notice, number three, feature in prophase is that the centriole have separated and they have taken position on the two opposite poles, and as they are separating, these fibers, which are made up of microtubules, these spindle fibers, that is number four feature of prophase, you are going to see that these spindle fibers are forming between the two centrioles that are separating and taking up place in the two opposite sides. One will be here and one will be here. Okay. All right. The next phase. So, I wanted to mention also that in a plant cell, there are no asters and there are no centrioles. So how does this microtubule form? Because I can see microtubules. So of course the microtubules are formed during the prophase, and these microtubules are formed originate from the MTOC. So this is the microtubule organizing center, which is located all over the nuclear region or the nuclear envelope, they originate from the nuclear envelope, which is breaking down, but these are centers that are present inside the nucleus, okay, in plants, not in animals. All right, the second phase is metaphase. So in metaphase, remember the word M for middle. So here, if you remember, the spindle fibers have come, and what they do is they attach to the centromere, this point. And the spindle fibers, like a puppet, the strings of a puppet, will pull the chromosomes, because now there's no longer a nuclear envelope. So the chromosomes are pulled and aligned in the middle of the cell. So keep this in mind, M for middle, M for metaphase. So metaphase is basically the chromosomes line up along the middle of the cell. All right, now here is the anaphase. In the anaphase, the chromosomes, the spindle fibers, they start pulling at the centromere. So the two sister chromatids, are going to be separated, okay? And these are going to be separated, and they're being pulled to the two opposite poles by the spindle fibers. So obviously, the attachment of the spindle fibers to the centromere is crucial, essential for this split to take place. So notice also that these are sister chromatids. When they separate, we call them chromosomes. So these chromosomes are now V-shaped, V as in Victor, and they're being pulled to two opposite poles of the cell. Now, the last one is, of course, the telophase. T stands for tail. So the telophase, what happens is just the opposite of prophase. And what happens is now the chromosomes are going to become uncondensed, that is 1. They are no longer going to be condensed, they are uncondensing, number 1. 2, the nuclear membrane is appearing, this starts to appear, the nuclear membrane reforms. And the spindle fibers, that's number 3 feature, spindle fibers are breaking down, no longer needed. And one more feature that should have been mentioned was that the nucleolus starts to appear, okay? So the nucleolus starts, you can see it, nucleolus reappears or reforms. Now, after the telophase is cytokinesis. Cyto means to cytoplasm, kinesis is to divide. So here in an animal cell, a cleavage furrow forms, and as you can see, as the nucleus is reforming, you are going to have this cleavage furrow. The cells are going to be pinched in the middle, and you end up with 2 daughter cells, each having identical chromosomes, and so this is in animal cell. But in plant cell, it's going to be a little different. Now, in a plant cell, they cannot pinch because they have a thick cell wall. So how can they do that? Well, what they do is that the Golgi now produces these vesicles which have the cell membrane and cell wall material, and these vesicles, with the help of microtubules, they move along the middle of the plate. Now the vesicles, notice, fuse, they fuse, they gather and they fuse, and what this forms is a cell plate. Now, the cell plate, what happens is forms first a cell membrane, and then what happens forms a cell wall. So cell membrane is formed, a cell wall is formed, and therefore you will end up with two cell walls, which later can become thick. So yes, with this we come to the end of our presentation. I hope this was helpful to you. If there are any questions or concerns, please call or contact me. And thank you for your time and attention.